Hi everyone, I am Maria Rodriguez, a master's student in the Katie Wagner's lab at the University of Wyoming. In my thesis, I'm working with Yellowstone Cutthroat Trout populations of the Teton River. And although I cannot join you in person, I am very excited to share with you the project that we are doing in Wyoming along with friends of the Teton River. First of all, I want to introduce myself. I am a biologist from Colombia. And coming from the highly biodiverse tropics, I am interested in understanding how biodiversity is formed, maintained, and especially how we can conserve it. During my time here, it has been wonderful to learn so much about the biodiversity of the Western US, especially about fish. The region's fish fauna is incredibly interesting, and it faces important challenges and opportunities. An example of this is what is happening with Yellowstone Cutthroat Trout, which is a native western subspecies of great cultural and economic importance. Unfortunately, the populations of this subspecies have shown a persisting decline throughout its distribution. This map shows the historical distribution of populations in light blue, and we can see that it used to be much wider than the current distribution shown in dark blue. We know that different human actions have been largely responsible for these population declines, and a very common practice in freshwater ecosystems has been the construction of dams. The problem is that these dams act as barriers that fragment the previously continuous distribution of populations, leaving disconnected populations of smaller size that are more susceptible to local extinction. We can think of this as a big problem for fish if dam limits the movement of individuals along the river, which is essential for maintaining large and connected wild populations. This is what I'm studying in the Teton River, where Phil Dam was built a little more than a century ago and disconnected the river in two sections. Although the dam has a fish passage, there is uncertainty as to how efficient it can be in allowing the movement of an adequate number of fish to keep the downstream and upstream fish connected. In order to understand how fill dam has affected cutthroat in the river, I am using genetics. You may be wondering why genetics? Well, the reason is that we can study the effect of barriers on populations based on the patterns of genetic differentiation they exhibit. When individuals can move freely between populations, these populations will be connected and tend to be genetically similar. However, the movement of individuals will decrease when barriers fragment the population distribution. Thus, populations will become isolated from each other and will become genetically differentiated. So after this brief background, I want to share with you some of the results that I have found so far. First of all, we collected genetic data from several populations across the river to have a good geographical coverage for a final dataset of 873 cutthroat genetic samples. With this, the first thing that I did was to explore the genetic variation across all the samples. The image that I'm showing you here represents an analysis that summarizes the major genetic variation among samples on two axes. Each point represents an individual sample, and the closer the points are, the more genetically similar they are. What this analysis shows is that, although there are some exceptions, fish upstream of the dam, shown with blue dots, are more genetically similar to each other than to fish downstream. Most upstream samples are clustered at the bottom of the graph. Likewise, fish downstream, shown in red, are more genetically similar to each other than to fish upstream. Most of them are clustered at the top. This means that at the global level, the genetic differentiation and genetic similarity is partially determined by the location of fish relative to fill dam. Then, I examined in more detail genetic differentiation across populations by asking how many differentiated genetic groups of cutthroat can be identified in the river. Before sharing what I found, I will first explain how to interpret these results. I will show you this type of plot where each vertical bar is an individual sample, and each color represents how many different genetic populations are identified. In this case, we have 5 colors, so 5 genetic populations. The color proportion of each vertical bar represents how much of its DNA belongs to a genetic group. For example, these 3 samples are part of only the green group, and all of these samples are part of only the blue group. Sometimes samples like this one will show more than one color, which is often interpreted as hybrid individuals. That said, the analysis found that globally, populations below and above the dome can be identified as genetically different, shown respectively in red and yellow. This indicates that the main population genetic differentiation coincides with the location of the dome. When analyzed at a finer scale, the analysis found that there are 4 genetically distinct groups throughout the river, which are shown in red, yellow, green, and light blue. This map shows where these populations are located. The red and the yellow are downstream of the dome, and the other 2, shown in green and light blue, are upstream of the dome. Therefore, this means that other factors have also shaped the population genetic differentiation in the river, which is something I am exploring in my work. Finally, I evaluated the degree of genetic differentiation among these populations, which may suggest how the dam has affected the movement of individuals along the river. This table contains values indicating how differentiated 2 populations are from each other, with higher values indicating greater differentiation. I found that the greatest genetic differentiation occurs when comparing populations that are separated by the dam, whereas the lowest levels of differentiation occur between populations that inhabit the same section of the river. Thus, this suggests that movement of fish between populations may be limited when there are physical barriers separating them. An important next step in my project is to test more explicitly whether fell dam was the cause of the genetic differentiation between populations below and above it. An exciting thing about genetics is that it allows us to trace events that happened in the past. Therefore, I will perform some analysis to estimate when these populations diverge genetically. And knowing this, I will be able to compare if that time coincides with the construction of the dam, and thus evaluate if it was responsible for originating the different populations that we currently see upstream and downstream of Fell Dam. I hope to have shown you with all of this that genetics is both a relevant information source and an innovative tool that is giving us novel perspectives to address conservation issues. Understanding how different human actions have affected threatened species such as cutthroat will be fundamental to take informed management decisions. And I would like to end by thanking the funding that Friends of the Teton River is providing me to pursue my graduate studies at University of Wyoming while I'm conducting this exciting project. My professional objective is to apply genetics for conservation purposes, so this is an incredible opportunity for me to fulfill this goal. Thank you all for listening to me and for your support to make this project possible.